Hello, welcome to the Chapter 46 lecture covering microbiology and disease. This is a lecture that goes with your 219 lab techniques class. In this chapter, we're going to be dealing with all things microbiology, so anything that we would collect a specimen for to view underneath a microscope and identify for treatment purposes. So during the microbiology process or specimen collection, what we're going to be concerned with was microbiology. So the study of microorganisms. This means both harmful and helpful microorganisms. A good example of a helpful microorganism would be resident normal flora. Resident normal flora is present in the body, has vital functions. It does not cause disease in most cases. It is helpful in areas such as digestion. And then we also have pathogens. Pathogens are the opposite, and these are going to be microorganisms that are going to cause disease or infection or inflammation and are problematic and need to either be helped with medications or treatment or need to run their course. Our body has a response to remove pathogens whenever necessary. Part of what we're going to be worried about within our area of medical assisting is collecting specimens for microbiologic testing. Whenever we collect our specimen, we are always going to store it and prepare it for transportation in a manner that's going to protect the integrity of the specimen. That way it can be sent to the lab to be processed and looked at for examination to be identified and treated. Microorganisms, if they're pathogenic, cause diseases in a couple different ways. We are able to not worry about many of them because they're not harmful. They're not a problem. They can exist within us and around us and not be problematic to us. We have a variety of defenses that our body conducts on its own. For example, a fever. A fever is your body's way of creating an environment that is not ideal for microbiological growth. So it's making an area too hot to where it is not a condition that a pathogen enjoys living in. We also have to have conditions that are favorable for a pathogen to grow. For example, if we have small children and they get sick frequently, which they do, that is because their body has not developed immunity yet. So it doesn't have any type of antibodies to fight a recurring illness that maybe is experienced before, whereas an adult who maybe has had the same pathogen in their system before can either fight that off and be minimally ill or not ill at all. So that's something that we look at. This is also why we do vaccinations. Vaccinations introduce a small dead version of a pathogen so your body can create an antibody. Essentially, what an antibody is a little bodyguard that's made to remember pathogens that have been in your body before, which is why some of the tests that we conduct in lab techniques is looking for antigens, that is the actual problem-causing germ that is generating a response, versus an antibody. An antibody tells us that we have or have had that antigen in our system. We have to be able to avoid defenses to be able to cause diseases. So we talked in 110 about breaking chains or cycles of infection. So that means that we're washing our hands, we're stopping it there, that we are taking medications, that we have proper nutrition, that we are getting proper rest and low stress levels, and our body is performing at an optimal level to be able to fight off the different microbiologic or pathogens that might enter our body. So as long as we are able to intervene, we won't set up with some kind of disease or illness. If not, it is going to set up, replicate, and then we're going to get ill. Microorganisms and diseases mechanisms. Microorganisms work in specific ways. Not all of them cause disease the same way. Some of them actually have various mechanisms with which they perform their destructive behaviors. So some microorganisms will consume nutrients that our bodily cells need, so it cannot do what it needs to function. Sometimes microorganisms will get inside of cells reproduce with inside cells or deform cells and cause them to replicate, and now we have abnormal cells that are producing. Sometimes the cells or the microorganism can cause our body to target our own defenses. Autoimmune diseases are a good example of this. So our body will attack its own things that is not necessarily meant to function that way. Also, some microorganisms will produce cell or tissue damaging toxins, so it will poke holes in our cells and destroy them and break them down, causing us not to work as efficiently as we are intended. Our microorganisms are broken down and classified into a few different categories. We have subcellular, which means there are no cells on this one. It is literally pieces of material, such as DNA, RNA. Viruses are this. Viruses do not have a cell of its own. They're very small. And they don't have any kind of organelles or nucleus or anything within it to be able to function independently. We have prokaryotic microorganisms. These are very simple. So pro means one. There's a single chromosome. They have no nucleus and no organelles. These are what bacteria are. This is a classification of bacteria. And then we have eukaryotic. Eukaryotic are a little bit more sophisticated and complex. They're highly structured. They do have a nucleus and cytoplasm and organelles. So protozoans, fungi, and parasites. So think of bugs and worms and a little bit more of a sophisticated microorganism versus the simpler bacteria or even subcellular viruses. We have a different kinds of pathogens. So not only just thinking about microorganisms helpful and harmful, but specifically pathogens harmful. So we have prions. These are made of proteins. It's the cause of several different illnesses. Very small, no nucleic acid, don't really know how they reproduce. Even smaller than viruses. Then we move up to viruses. Viruses, again, are DNA or RNA that are wrapped up in a protein coat. They have to be inside of a living cell to reproduce, so they're a little bit of a freeloader, honestly. Chickenpox is an example of a virus. Flu is an example of a virus. Bacteria are, again, single-celled, so prokaryotic. They reproduce quickly. Asexual reproduction, so they don't have to have any other kind of reproductive source other than themselves. Cholera is an example of bacteria. Protozoans are very similar to bacteria, but they are larger than bacteria. Dysentery. Typically, protozoans aren't extremely common, but they are the leading cause of death in developing countries because they're usually found in contaminated water and soil and food sources because there isn't open availability to hygiene capabilities. Fungi are multicellular. Think of budding, think of the stuff that grows on your food in the refrigerator, thrush, people that have like toenail fungus, things like that. Typically, you have to have a weakened, a little bit of immune system for fungus to be able to grow. There has to be some sort of condition that makes it optimal for that to be able to happen. And lastly are helminths. These are parasitic. They have organs. So worms are helminths. So these are all disease causing. They have to get into your body, so they have to get through a chain of infection to be able to get in and set up shop to be able to reproduce and cause an issue. Viruses are one particular area that we just talked about of pathogens. Viruses are so small that you cannot see them with a regular microscope. They're simple, simpler than a cell. Remember, they don't have a nucleus. They have to reproduce within another living cell. HIV and hepatitis are two very significant bloodborne pathogens, pathogens that we are worried about. Viruses there's a large list here. Some common ones you're going to hear a lot about is influenza. Influenza is different. So the flu is not your stomach flu. Think about flu as in it is going to hit you like a train. Like it comes on fast, usually has a fever, body aches, headache, just miserable feeling with some respiratory symptoms. It's not something that creeps in slowly. When we test for flu, we test for flu A and B. Very similar. However, flu B replicates slower than flu A. So if you can get antivirals in early, flu B is going to be easier to kick than flu A is, but they do have similar symptoms. Hepatitis is another virus that we think about. Hepatitis is going to affect your liver. It's going to cause some nausea, vomiting, a little bit of abdominal pain, jaundice, so you're going to appear yellow. Very uncomfortable. We also have mono is just something that we're going to test for in lab techniques. This is caused by the Epstein-Barr virus. Sometimes mono goes undetected because we'll sort of check everything over top of it first, but it has a fever, a sore throat, swollen lymph glands, usually a lot of fatigue. Some people note nausea associated with mononucleosis. And here's a chart with some additional viruses for you to review as well. And even more. Chickenpox, by the way, is a virus. Chickenpox and shingles get confused a lot, so I like to clarify this a little bit. Chickenpox is caused by the varicella zoster virus. Whenever you turn, I think it's 50 or 65, you can get a shingles vaccine. So basically, you cannot get shingles if you have not had chickenpox. So somebody who has either had the vaccination or had active chickenpox in their life has the varicella virus laying dormant in their system. Sometimes conditions become favorable and that virus will reactivate and create it feels like a tingly, prickly sensation and a rash that will blister and leak fluid. The thing about shingles is it will not cross midline. So if it's on one side of your body, it will not cross over the center line of your body. Is a good way to be able to identify if that's what you suspect you have. Now, if you're a person and you have shingles, shingles is contagious, specifically by the particles that's in the fluid of the blisters of shingles. So it's important that those are kept covered and not contaminated to other people. We want to stay away from people that are immunocompromised or pregnant until the rash has dried up and crusted over. If you do come in contact with somebody, they cannot catch shingles from you, but if the person is susceptible to chickenpox, you can give someone chickenpox from your shingles outbreak. Bacteria is the next type of pathogen we're going to talk about. The good thing about bacteria is typically if we can get a sample and identify what type of bacteria it is, it's really effective on being able to get the right antibiotics and give the person medicine that's going to make them start feeling better quickly. We do this a couple different ways. We collect a specimen and create a culture, and off of that culture, we can identify what the bacteria is. And then we add the medicine we think is going to work to the petri dish that we have grown the bacteria in. And if it creates a little halo or kills the germs around it, we know that that is the drug that's going to be effective. So that is called a culture to collect and grow the specimen. It's called a sensitivity to add the medicine to it to verify what it's going to be used for. Now, when we have the specimen for our culture, we're going to grow it on a petri dish, and then we are going to put it on a slide and put it underneath a microscope and take a look at it to be able to tell what it is. And we do this by a couple different things. First, by what kind of symptoms and what bodily system it's affecting. Then we're going to stain. We stain the specimen so it's able to be seen underneath a microscope, as well as, depending on what color dye the bacteria holds, it lets us know what kind of a cell wall it has. So one of the things that we look at is the shape of the bacteria. So we have the cocci, which is a circle or a sphere. Looks like a grape kind of. Bacillus, which is a little like oval, sort of like a tic-tac shape. Spirillum, which is a corkscrew. Vibrio, which is like a curved bacilli, but it's basically like a comma. And then we look at how they grow. Do they grow in sets of two? Do they grow in clusters? Do they grow in chains? And that's going to tell us more about what we're looking at. So If it grows in a cluster, like grapes, that would be staph. So staphylo means it's growing in clusters or groups. Diplo, think about dice is two, is they're growing in pairs, so sets of two. And strepto grows in a strip or a chain. So strepto, whatever, would be a strip or a chain of whatever shape. So it might be streptococci. Those are things that you'll see. We also look at the ability to retain stains. So whenever I told you we take the cells and we put them on a slide, we're going to put that over a heat source, usually open flame. That's going to bake and adhere the specimen to the slide. And then we're going to put different stains on it. We're going to apply a blue stain, rinse it off, red stain, rinse it off, iodine, rinse it off. And then whatever colors show up is going to tell us what type of cell wall it has. We call these either gram positive or gram negative. Gram positive is purple, or they'll look bluish, like dark blue. So positive, P for purple. Gram negative are red. Some bacteria will not take either stain. That is because the outer cell is waxy, and so it just doesn't hold. It just rinses right off. For that, we have to use an acid-fast stain, which is going to break down the cell wall a little bit and allows us to view. Tuberculosis falls in this category. So TB has a waxy cell wall. We're also going to look, when we do our culture, did the cells grow better in a container that has oxygen or does not have oxygen? So an aerobic bacteria means it likes oxygen. Anaerobic means it prefers environments lacking oxygen, which is usually what we get on the inside of our body, because you don't need oxygen inside your body. If it can grow in both, it's called facultative. So some will do both, and that just kind of helps us to narrow down and identify what cells are there. We also look at biochemical reactions. So we look at what kind of environments the bacteria like to grow in. So our different petri dishes aren't all the same. Some have blood in them, some have broth in them, some have seaweed in them, different things that have different kinds of nutrients that will fuel or feed different bacteria and help them grow better. So that's another way that we're able to identify what kind of cells we're dealing with. Here are some bacteria as well. So these are bacterial infections. These are things that if you go to the doctor and we can do a swab. So when we do like a strep swab or we do a flu swab or things like that, that helps us to identify specifically what is going on so we know what kind of medicine will work. Because if we try to treat your flu with an antibiotic, it's not going to work because it's a virus. So we have to know what the problem is so we're giving you the appropriate medicine. Sometimes when you go to the doctor, they will give you a general like antibiotic to try to help kind of tamp down symptoms until your culture gets back, because a culture takes about 3 days. Sometimes we have patients that take it and it does help with some kinds of bacteria that might be in there, but it's not killing the predominant issue. So as soon as you stop taking that antibiotic, if you're not switching to the one that's correct, then everything's going to come back with a vengeance, and you don't have the normal flora in there anymore to help fight off and tamp down symptoms. So it is going to be a raging infection if you do not switch and get the medicine that's effective for your problem. Bacterial infections. So some of these could be chlamydia, Lyme disease. Fun fact, koala bears are super cute, but they're infested with chlamydia. Whooping cough, anthrax are all parts of bacteria. You could also are caused by bacteria. Also, Legionnaire's disease, meningitis, Rocky Mountain spotted fever, which comes from tick bites. Lots of different kinds of illnesses. But again, if we can identify the bacteria, we can identify what medicine is going to be effective in treating that disease. Shingles, or shingleosis, bacterial pneumonia, syphilis, strep throat, cholera again. There are some special groups of bacteria that don't behave as most do. For example, TB and leprosy have that rod shaped waxy cell that would require an acid fast stain. Rickettsia, which is rickets, these live on other living cells. They're found in ticks and mites. So they will cause Rocky Mountain spotted fever, which is a rash that will be all over your body, including your palms and the soles of your feet. Chlamydia, which grows on other living cells as well. It's actually an STI. It can cause eye disease, pneumonia, and some heart diseases. And then mycoplasmas. These don't have a rigid wall, so some STIs and pneumonia fall in that area. We have bacterial pathogens because bacterial pathogens set up and make you sick quickly because there's a lot of them and they're the main cause of disease in humans. So that's why when we go to the doctor, a lot of times, if you go in and you tell them that you're having symptoms in the onset and how it happens, they're determining if you probably have a bacterial or a viral infection. We can't just give antibiotics to somebody if we don't know a cause, because what will happen is we create these super germs that will not respond to antibiotics any longer, and then we're going to have a major problem later on when we have no medicine that we can actually treat our illnesses with. We also have drug resistant microorganisms. Think about MRSA. MRSA actually lives in most people's noses, which is fine as long as it's there. But if it comes through and it gets into somebody's, like a sore or a wound, it will set up an abscess, and that abscess is resistant to normal antibiotics that we would treat an abscess with. So we have to identify what is inside of it. That way we are making sure we are giving the correct antibiotic. And the antibiotic for MRSA is strong and it smells really bad. And if you're taking it orally, it isn't a flavored antibiotic. So sometimes we'll have people like take it and then like mix it with chocolate syrup in a syringe and like just choke it down the best they can, because it is pretty nasty. Some risk factors to make you susceptible to being affected by bacterias, pathogenic bacterias, would be your age and severity of illness. This is why most people in the age range of 65 and older are considered high risk, because that's the age where they've determined majority of people's immune systems start to decline and not be as effective as they used to be. Invasive procedures. So anytime we break the skin or make a direct path to the blood or tissues, we're creating an environment that would be easier to infect with. The use of antimicrobial agents, so using too many of these things and not letting our body create its own defenses. Repeated contact with the healthcare system. It sounds counterproductive, but there are high concentrations of sick people and germs in hospitals, which is why used to, when you had like a hip replacement, you would be in a hospital for weeks. Now we try to get you in and out and away from the hospital where there's germs and more potential for contamination. And then underlying disease and conditions. For example, people with diabetes aren't as likely to heal quickly and efficiently. So we want to make sure that we are treating people and keeping all of their illnesses and disorders in check. That way their body can function at the best of its ability. To help prevent antibiotic resistance within the healthcare system, we want to avoid infection, so which is why we practice our aseptic technique or our surgical technique. We want to improve prescribing antibiotics for correct illnesses, and to make sure the person's using it all the way. A lot of people will take antibiotics so they feel better and then they quit, and that's a problem as well. We want to improve diagnostic testing, make sure that we are identifying what we're dealing with, educating patients on how to use antibiotics, reducing antibiotics in long-term care facilities, and then limiting antibiotic use in food and animals, which is really tricky because, like it or not, if we pump hormones and antibiotics into our animals or our livestock or our meat sources, which is what we do because we don't want them to get sick and take out a whole herd of beef or chicken, we're going to ingest them. There's a reason why 7th graders don't look like 7th graders from way back when. We are taking in things indirectly that we don't realize. So there's a lot of people that are become more conscientious of that and buying organic or certain types of meats from certain providers to limit the amount of antibiotics being ingested. Protozoans are the next pathogen we're going to talk about. These are single-celled eukaryotic organisms. So these are like bacteria, but fancier. They are larger than bacteria. Again, leading causes of death in developing countries, found in the soil and water. Most don't cause diseases in people. They can be more damaging to animals. This is also the cause of malaria, dysentery, and trichomoniasis vaginitis, which was trick. Really dangerous to immunocompromised people because they're an opportunistic type of infection, and they're going to be able to take over quickly. More protozoans on this chart if you would like to read in and look at the specific symptoms and causes. Next, we're going to talk about fungi. So these are eukaryotic. They cause fuzz. They cause strange growths on different items, have a rigid cell wall. They're tricky to kill because we can take one kind of medicine and treat it topically, but that's only going to kill the budded parts of the fungus. If the things are still haven't produced yet, then they're sort of like lice where they can sit in and they can withstand the treatment. So usually people have to take an oral. Antifungals, antiparasitics are really hard on your kidneys, so repeat treatment isn't really something that doctors will do a lot on those. Mold produces spores, which are hard to kill. These grow into large, fuzzy, multicellular, complicated organisms. This is the cause of athlete's foot, ringworm. I know a lot of people hear ringworm and think it is a worm. It is not. It is a fungus. Thrush, and vaginal yeast infections. Next, we have multicellular parasites. So these are bugs and worms. Really gross sounding, but to be honest, if we understand how they work, we're going to be more effective on educating patients and ourselves to treat and eradicate them. Roundworms and pinworms are found in your digestive system, frequently from undercooked meat. Have had patients who share food off their plate with their pets, and that has created different kinds of worms. If a person has pinworms, frequently they'll have rectal itching, spreads like crazy. So typically if one person in your house has pinworms, everybody's going to get treated, like without even seeing a doctor. Everybody needs to take medicine. Tapeworm has to be identified as well. Making sure that we cook all of our meat, we clean and don't cross-contaminate is going to be the key in not spreading these. Parasitic insects, so we're going to think ticks, lice, mites, and mosquitoes. Not only are they a pain, but they can spread disease. Fun fact about lice right now, schools can no longer send children home from school with lice because it is not necessarily contagious or problematic. It is a nuisance. It's not considered a disease. So if you want to protect your kids, if there's a lice outbreak at school, check their heads frequently. Lice do not like dirty. They like clean hair. So if you put in some like gel or some hairspray, things like that, that'll kind of help deter a little bit as well. Pull hair up and away. Don't leave long shaggy hair down to spread. Have your kids put their coats in their backpack or something like that and they're not in shared communal spaces. If you find your house does have lice, call your doctor, get some special shampoo, make sure that you use it several times because again, you got to kill what's in an egg and what is not. Clean all of the bedding, all of your animals, clean your stuffed animals, things like that. It is a not fun thing to deal with, but it is treatable. So how do we diagnose infections? First of all, we're going to examine the patient and look at the area and see if it's localized or systemic. We're going to get a specimen. So anytime somebody comes in and they've got like a wound or something like that, we're going to take a Q-tip and collect it and try to grow it. So the person comes in and says, I have this wound, it's sore and it's hot and it hurts and blah, 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 but it's scabbed over. We're going to cut that scab off because we need to make it ooze so we can get some of that pathogen holding specimen. We're going to culture the specimen by spreading or inoculating the specimen onto a petri dish. That's going to go into an incubator, which is a human body temperature for several days. The purpose of that is to create a body's environment and see what'll grow. Then we're going to identify the sensitivity, so culture and sensitivity, and then the doctor will get those results and order how to be treated. When we collect specimens, we want to make sure that we're using devices that are sterile, so we're not picking up pathogens from outside the environment. So it needs to come from sterile packaging. Anything that's sterile is going to have an expiration date on it, so let's make sure that we're checking for those. They get put into collection or transport systems that is going to protect the specimen, not add anything to it, not break anything down within it. We're going to collect the specimen by the correct guidelines. So, for example, if a person has strep or something weird in their throat, you'll hear a lot of people say, like, I can't have strep, I don't have tonsils. No, but you might have some kind of bacteria growing in your throat causing an issue. So we're still going to collect a specimen there. So we're going to swab a throat. If the person has urinary tract symptoms, they're going to get a clean catch midstream, and we're going to grow that. So we have to teach them how to collect correctly. Sputum specimens, so if you have tuberculosis, you're going to have to get some of that mucus up out of your lungs. So we're literally going to have the person cough and hack up as much mucus as they can and spit it into a sterile specimen cup. Usually, frequently we can use like a urine cup. And then wound specimens, we're going to use swabs that go into a tube that has a some kind of preserving agent in it, and that way it can be sent off as well. Our transporting agents have to be able to be labeled because we have to put the patient's identification on there. It's also going to create an environment where it's going to stop the specimen from growing or deteriorating, and to protect whoever is moving it from an exposure to infectious material. Think about this. If you are a person and your job is just to transport, so you might be the courier that goes around to the labs to get specimens to bring it back to the lab, the main lab. If you were to drop a container and your specimens fall out, or if you are housekeeping and you are cleaning up different trash cans, we want to make sure that if something were to bust through, I should not be exposed directly to somebody's infected bodily fluids unknowingly. So we want to make sure that we are putting all of our stuff into cases that's going to be double protected. That way we're not hurting our coworkers. There's usually daily pickup from outside laboratories. So there's somebody that drives around, know when that last pickup is, so you can help store or get it in as you need to. Sometimes we have to mail in specimens. If that's the case, there are specially designed packaging that identifies that has a etiologic material in it. So like say that there's a UPS semi wreck on the interstate. If there are items on the highway that's contaminated, again, that needs to be able to be identified. There's a couple kinds of specimens we collect that are direct examination. So we are going to have to get the specimen and get it to the lab so it can be viewed immediately, because we want to keep whatever is in it alive because it helps us to identify. So one of the ways we do that is with a wet mount. So we use sodium chloride or saline to help mimic the wet environment that the organism came from. So trichomoniasis vaginitis, that one is actually a motile, which means it has like a little tail. It swims and jerks around. So whenever we collect that, we have to collect it, which is a pelvic swab, and then we're going directly to the lab with it in saline and putting it underneath a microscope to identify it, and you will see it swim, like it moves. Sometimes we do KOH mounts. These are potassium hydroxide. KOH is just like the anatomic table version of that, periodic table, sorry. These are used for fungal infections. So we use a special formula of solution to preserve the fungal infection to identify it. It does have to sit for 30 minutes before it can be looked at, because it has to dissolve the outer keratin of the fungus before we can see. For preparation, again, I talked to you guys about stains a minute ago, but this is the specific way that it works. We dry and fix with heat, so we put a smear on the slide and then we heat it up. We're going to put on purple stain, which is called crystal violet. We're going to put on iodine, which is a mordant, which means it locks it in. Decolorizer, so we're going to rinse that off. And then we're going to apply red and then rinse that off. And if it's purple, it's positive. If it's red, it's negative. And if neither of those work, we're going to have to do an acid-fast stain. Whenever we are doing cultures in the office or growing microorganisms, we have to choose what our base is going to be based on what we think the organism is. Blood agar happens the most common because that's what most bacteria in the human body prefers to live on. We always label on the plate near the edge. We don't put it on the lid because it's easy for us to take off multiple lids and now we don't know which one it belongs to. Whenever we create a smear, when there's clusters of bacterial growth, we call that a colony. And our agar gelatin, is the gel that's inside of a petri dish. That is made from seaweed. Whenever we create a smear on a plate, that's called inoculating. Sometimes we do a qualitative and a quantitative analysis. So remember, qualitative gives us a positive or negative, yes or no, is it there? Quantitative is going to tell us how many are there. It's literally somebody's job to look through a microscope and count cells. There are specialized slides that are gridded off, so it's easier to keep track of where you've been and what your pattern is. Incubating culture plates happens at body temperature with the agar side up. That way, whenever it is cooking, it doesn't cause condensation that's going to drip back into the specimen. And that is our microorganism lecture. I hope you guys learned something, and I will talk to you next class.